distinguished career and made great contributions to entomology. I'd first like to thank the prize committee, chaired as in previous years by Rick Foster. Where's Rick? I saw Rick. Oh, there you go. In my blind spot. So thank you to all who served on that committee. So this year's Osmun Prize winner is Dr. Alberto Ferreres from Spain. He did his undergrad at the Polytechnic University of Madrid, finishing in 1983, and then followed it up between 1984 and 1988 by a PhD, also at the Polytechnic University of Madrid. In the middle of that, though, he found time to come here for 2 years, from 86 to 87, to complete a master's degree. He then followed that with a postdoc at UC Riverside, working with Tom Perring, and from 1988 onwards was a researcher, initially a researcher back in Spain, initially at the National Institute of Agricultural Science, and from 1990 onwards at the Center for Environmental Science within the Spanish Research Council. He rose rapidly within that organization, becoming the director from 2009 to 2011, and was also the director of the Institute of Agricultural Science within the Spanish Research Council from 2006 to 2011. He is currently full professor of entomology within the Institute of Agricultural Science in their Department of Plant Protection. Alberto is an expert in the feeding of sap-sucking insects such as aphids and their transmission of plant viruses. So as you're all aware, this is of tremendous importance to agricultural health, and he's been one of the most successful scientists working in this very large field in recent decades, publishing over 170 papers, 20 book chapters, and is currently serving as chair of the International Committee on Plant Virus Epidemiology, which is part of the International Society of Plant Pathology. So please join me in congratulating Alberto, our 2017 Osmun Prize winner.
Thank you very much. Well, just put the microphone on. Do you hear me? Yeah? Great. So thanks very much. For me, it's a honor and a great pleasure to be with all of you, and especially in this building, which was my place for studies in entomology. This is where everything started in 1986, 30 years ago. So I'm really happy to be here back again. Of course, this wonderful auditorium was not at that time, but I'm really happy to be in the same place giving the lecture. So I will start talking a little bit on how my stay in Purdue, how the classes I learned here influenced my whole career, and how this starting point at Purdue was shaping me in all the scientific achievements that I got during my whole career. I wanted to explain a little bit about the courses, the teachers I had here, how the master's thesis shaped also my future research, and also the collaborations that I had with different students at that time, which are still in touch with me when I started my career in Purdue, and how important it is to have international collaborations, long-lasting collaborations with many different researchers that are working on our field. And this opportunity that Purdue gave me 30 years ago has been probably one of the reasons of my success in this career. I joined the International Committee of Plant Virus Epidemiology in 1999, and now I am still the current chairman of this society and this working group. And also I will end out my talk talking about the different research lines which I have been running in the last 30 years. So let's start. So this is also one of the things I wanted to do today, was just to summarize everything that has been done, that I did during my 2 years in Purdue, And one of the persons that I really miss and that I really want to acknowledge is Truman Martin, who was the person that didn't know me, but he invited me to his house for 2 months until I was able to find an apartment in Williamsburg, and this was 1986. Also, I was able to rent a car, my first American car, beautiful Ford LTD, and an excellent experience of driving such a large car in Indiana. So those are the first things that I would like to remember, and one of my memories that I have from Purdue. Also, when I came here, we had there was a new aphid pest coming, the Russian wheat aphid, and at this starting point, I had some experience with this aphid in Spain. So my first publication at Purdue was about the Russian wheat aphid, and this was the starting point of some interesting studies that I did with aphids, several aphids, while I was here doing my master's. So I really remember that paper also, and I wanted to remember it now. Also the courses that I was taught here have been the background of all my work in the following years, and also the connections with international students, made excellent friends, and also I was able to make my own living. I was living with my family, with my parents at that time, so when I came to Purdue I had to learn how to cook, how to make my bed, how to do everything, and how to make my own living here. So this was also another good lesson that I was taking from my experience at Purdue. And of course the excellent courses and excellent professors, some of them are here, and I would like to acknowledge them also very much for the excellent lessons I received at that time. Some of them passed away, well, Bob O'Neill, but he also gave me very excellent lessons on my future career, as well as Richard Lister, which taught an excellent course on plant virology, which I have been using until nowadays. These are some notes that I took during these lessons, some of the quizzes that I remember from Alan York's course, who taught us many things, but one of the useful things was the names, scientific names, and common names of pests, which are an excellent way to know how to describe the pests and how to understand what were the host plants of these pests. And also these excellent lessons on key concepts on entomology, which I still use nowadays. Handouts, which were very popular at that time. Of course, there were no PowerPoint slides as this, but there were many handouts, and one of them, which is this transformation data analysis, I still use it in my own lab right now and give it to students. Then the other course I took, which was excellent, it was Entomology 512, taught by Tom Terping. He gave us excellent classes as well, and beautiful diagrams and figures on how you could understand and learn IPM, such as key concepts or economic thresholds, which I also use nowadays to explain to my own students. So this means that the lessons learned in Purdue, I have been using them all during my whole career, and that they were really impacting my whole way of understanding the entomology. These notes, I still keep them in my lab, and I still use them in my lab 30 years afterwards. Silent Spring was also another starting point of my interest in studying entomology, so thanks very much for that. And also, I have to acknowledge my advisory committee. My advisory committee was a very good blend of entomology, plant pathology, agronomy, all together. And I learned things very good interactions of what I am really doing today, which is plant virology and its connection with insect vectors of plant disease. So this master's studies was really good. This picture is 30 years old. It was in this same building, and actually this was the first studies I did here on the effects of barley yellow dwarf on the life history of the English grain aphid, Cytobion avene. And this was one of the studies I did for my master's, and studying how these infected plants were affecting the life cycle of the insects. And this is another good lesson and another technique that I learned here at Purdue by Richard Schuckels, who was a student of Bob McLean, who was the one that invented the electronic feeding monitoring system in the 60s. And Richard Schuckels had his own equipment here, which was made by Doug McLean, and he taught me how to use this equipment. He taught me how to wire aphids, which is really a tough and difficult thing when you do that for the first time. And this was a technique that I have been using in my lab for different studies on host plant resistance, on insect response to insecticides, on transmission of plant viruses, and transmission of plant pathogenic bacteria right now. This was the old equipment that he had. I don't know if it's still around, but this was in the basement of ARB building, and it was an excellent equipment, very good one. At that time there were no computers to record the signals, so we recorded the signals on paper, and we had to measure with a ruler the time spent by the aphid doing specific behavioral patterns. And of course now we are using computers to acquire all this information. This is the first talk I gave in an ESA meeting. Also, while I was in Purdue, we went to the ESA meetings in Boston, and I gave this talk in the Boston Hilton Hotel of part of my master's work. Here's this Richard Lister in my apartment in Williamsburg with some other friends. Two of them I still keep in touch. For example, Juanjo Manfredi, he's a professor of mathematics in University of Pittsburgh, or Jaime Araya, who is a professor emeritus at the University of Chile, and we still keep in touch. Well, the graduation ceremony, which is also was very special for me, and the master's thesis, which I need to acknowledge again to my brother Elias, who was the person that encouraged me to come to Purdue and to stay here and leave my family on the other part of the world. Of course, this is also a picture I found from that time. I had hair by that time, so it looks very different from now. And the master's degree, which was an excellent experience for me. And to celebrate this master's degree, we went together with my friend Jaime to the Niagara Falls and to New York by car. And so this was the trip I had in mind after I finished my master's thesis. So then, how did Purdue shape my goals? Well, after I came back to Madrid, to Spain, I wanted to do a postdoc. After finishing my PhD, I went to Riverside and worked with Tom Perrings, also in the subject which was part of my studies in Purdue, which was how did the environmental factors alter the persistence of viruses in the aphid vectors, because one of the questions we wanted to answer is how long the aphids could remain viroliferous and how environmental factors would affect the persistence of the virus in the vector. And for doing this, I moved to Riverside, which was also a great experience for me. Also, collaborations over the 5 continents have been very successful and have been a really wonderful experience. And I had friends and colleagues from many different countries. This is Stefan Blank and Frei Talingi, which both of them work in Europe, and we had excellent collaborations with them. And I also wanted to highlight my sabbatical leave in University of Urbana-Champaign with Mike Irwin, where I spent 1 year working on aphid movement and aphid preference for infected plants. And we are also good friends, and we still keep in touch. During my stay in Mike Irwin's lab, I also came back to Purdue, and I spent a weekend with Truman Martin in his house. And at that time, I was already married, and I came with my wife and my children, my son, as you can see here in this picture, in the country fair at Purdue in summer 1997. So Merche, who is here with me, she came to 1 year to work here and to come with me and spend 1 year in Mike Irwin's lab, and we had the chance to be in Purdue. And this was my last time in Purdue until today. So good. I also wanted to speak a little bit about the International Committee of Plant Virus Epidemiology, which also has helped me a lot during my career. I became a member in 1999, just after I came back from my sabbatical. And this committee, which was created by Mike Thresh from the UK, served us to put together plant pathologists and entomology to work on plant virus epidemiology. And I really liked the working group, and I really joined this committee from there until today, because it's the only committee, the only part of the Society of Plant Pathology where both entomologists, plant pathologists, molecular biologists, epidemiologists come together and celebrate meetings on what I am really interaction, which is with the interaction between insect vectors and plant pathogens, which is my major interest and topic, which I think needs a working group, and I found this working group in this part of the society. So I was invited to organize a meeting in Madrid, in Spain, in Almeria, in the southeastern part of Spain, and at that time I made excellent connections with different researchers. We had very good discussions with different people at that meeting, and this also helped me to interact with many different professors and researchers all over the world. I also wanted to talk about the expeditions to Africa, because at that time, after I came back to Europe, we had European Union projects to work on Bt crops and the persistence of Bt in the soils, but also on how clays could be used to increase the persistence of Bt israeliensis to improve the mosquito control. So we conducted some experiments in Africa, and we looked at the persistence of Bt clay formulations to see how this could be used to control mosquitoes. So this was also an excellent experience on how we could use entomology to reduce not only yield crops but also save some lives. I went back to Tanzania recently in 2013 for another meeting of the International Group on Plant Virus Epidemiology, and this was also an excellent experience for me, and at that time I was already a chairman of this working group. Also, I wanted to highlight the important thing, which is a long, stable international cooperation with Brazil, which I spent several visits since the year 2000, and we still have very good connections and exchange of students with Brazil. And actually, I went there for a short sabbatical spread in 2 years, where I conducted my own experiments on the influence of volatiles on the behavior of whiteflies when they are infected with different plant viruses, looking at what was the response and the coevolution between viruses and vectors. Training and exchanging students. That was another excellent opportunity in Brazil, where they were very interested in our work, and we did several courses on EPGs. EPGs are the electronic probing, the electronic penetration graph technique, the one that at that time was called electronic monitoring system in the US, but it's the same technique basically. And these training courses on the EPG technique were held in Brazil, in several parts of Brazil. And we set up some labs which are using now this technique around Brazil. Actually, there are now 4 labs working on EPGs in Brazil. This was one of the last courses I taught in Viçosa, in the University Federal of Viçosa, which I know you have very much connections between Viçosa, because Viçosa and Purdue had a joint agreement where they had exchange of professors, and it's a university where I still go often to teach these EPG courses. And these are some of the students that attended the meetings, and some of them in my lab, and this is one of the international students from Brazil who was trained in my lab for 2 years on EPGs. You see, if you go to Viçosa, in Minas Gerais, you will see an avenue, which is called Purdue Avenue, inside the university. And when they showed me the university for the first time, I was really, well, this is my university. What's doing here, this sign? And they had their own street, which was called Purdue University. But not only that, this successful cooperative venture has a building with different plates, which I also found there when I was visiting. And one of the classrooms is called Purdue University Classroom. And they did a lot of celebrations at some point in 1997 to celebrate the participation of American professors in the project, cooperative project that they had with Viçosa. I think this project ended in 1973, but for many years they had very strong links. Then another aspect I wanted to highlight was that Australia, a country which I never visited, just for an international meeting of entomology in Brisbane. But then recently I came back to Australia because one of the researchers working there on how climate change may affect aphid biology and behavior invited me to give a couple of lectures, and I had the opportunity to visit the free air carbon dioxide enrichment facilities, which I think is an excellent way to understand how different environmental conditions may affect insects and pathogens under a climate change scenario. So we started this collaboration like 1 year ago, and I think it would be a successful collaboration for the future. Well, now, after this long introduction, I would like to explain a little bit more about what I do, what are my major research lines, and what were my major achievements during my career. So, one of the aspects I wanted to study was how the viruses are transmitted by aphids mainly, and to understand how non-persistent viruses are transmitted by aphids. At the time we started to work, there was a hypothesis which was called the Ingestion hypothesis, which was proposed by Kerry Harris. And this hypothesis, to explain the transmission of these viruses, basically was based on some observations at the microscope where the insects, the aphids, were able to ingest the food that was previously ingested. But we thought that salivation could also play a role in the inoculation of viruses. So therefore, we did some experiments, which I will explain a little bit later, to show that ingestion salivation was probably the way that viruses were transmitted. Then I have been working on the coevolution of plant viruses and their vectors, how plant viruses are able to modify their behavior, and how this may enhance the own spread of the virus. So this has been also another subject of my studies, which I also will go over afterwards. And then also I've been working on how you can manage these plant pathogens, but not the pathogen itself. How could you manage and reduce disease epidemics by controlling insect vectors? And we have been doing studies on host plant resistance to vectors and physical and chemical barriers to prevent the spread of vectors. And finally, another area of research which I have been conducting is how vectors of plant viruses can respond to different environmental factors and different stresses. So this is the last topic which I will cover. To go over to how to understand and how to study the mechanisms of virus transmission by sub-feeding insects, what you can use is this technique, which is called now electrical penetration graph technique, or electronic monitoring system, which I learned here. And this technique has been improved in the last years to try to understand the behavior of an aphid or any other sap-sucking insect. And it's basically the insect is part of an electrical circuit. When the insect starts to feed, the circuit is closed, and any changes in voltage can be detected by the system, and you can actually know what the insect is doing, if the insect is salivating, if the insect is ingesting, and also you can correlate the position of the stylets within the plant tissue. And this technique is very powerful and can give you a lot of information for people working on feeding behavior. So what we did to understand the transmission of non-persistent viruses was to use this technique, and while you were recording the waveforms, you could identify specific waveforms like these ones, and you could remove the aphid at given points to know which type of waveform is associated to the acquisition or inoculation of a given virus. And this technique, we've been using it quite frequently to understand the transmission of the different types of viruses. And we were able, for example, to understand how these intracellular punctures, this is the EPG signals that you obtain when a plant cell is punctured, and as you can record this in real time, you could remove the aphid at a given point and see which of these waveforms is associated to the acquisition or inoculation of a virus. And for these non-persistent viruses, we found out that the common duct is probably the place where the viruses attach, because when you remove the aphid here during this 2-1 phase, you get a high inoculation rate, but no acquisition of the virus. But when you remove the aphid after it finishes up this third phase, you get the acquisition of the virus. So this, actually what you are using is a virus as a marker of intracellular stylet penetration, which is also called a potential drop. So you can know exactly in which part or where is or at which precise moment is the virus inoculated or acquired by the vectors. So with these studies, we proposed the ingestion salivation hypothesis, where we suggested that the virus particles were attaching to this common duct of the stylets, where the maxillary canal, where the food canal and the salivary canal fuse together. And therefore, the aphid is egesting or secreting saliva, which is flushing out the virus particles. For the ingestion process, it's the same thing, but what happens actually is that the insect is ingesting the virus, and the virus is attached to this area where it is retained by the aphid. So this is the ingestion salivation hypothesis, which still stands up today. And this work was conducted about 20 years ago in my lab. Actually, how this was proved is because another non-persistent or non-circulative virus, which is cauliflower mosaic virus, uses a helper protein, which has been detected, this helper protein attached to GFP, fluorescent protein, and it has been detected only in the common duct of the maxillary stylets when it was incubated, and this work, which was one of the collaborations that I had with Dr. Stéphane Blancs in France, showed exactly that the common duct is the hot point where all virus particles attach to the stylet. So actually the salivation ingestion, or ingestion salivation hypothesis was proved after these studies and some other studies that we did. Okay, so this is one of the contributions I wanted to highlight. Also, the work I did on the coevolution of plant viruses and their vectors, where we studied different systems, such as the BYDV Cytobium avene that I started here, where we did a work on how there was a mutualistic effect mutualistic relationship between the aphid vector and the virus. In this case, we wanted to do a similar study with Bemisia tabaci and how the tomato yellow leaf curl legomovirus could affect or could directly affect its feeding behavior. So for doing these studies, we actually used two different techniques. One was the EthoVision video tracking system, which allows you to record the movements in real time of an insect while it's feeding on a given place. So what actually happens is that you can track the movement of whiteflies, and for doing this we used two types of whiteflies. One was a whitefly that was infected with a virus, and you could also see how the whitefly moves around the petri dish, and you can actually measure the speed of movement, the distance moved, and you can also track exactly what are the directions that the whitefly is doing during his exposure. So we use two different sets of whiteflies, viraliferous whiteflies, and we compare them with non-viraliferous whiteflies to see if they could behave the same or differently. So what actually happened during this study, we actually found that infected whiteflies behaved in a different way as the non-infected. So you can see here in this slide that actually the infected whiteflies moved much less. They moved at lower speed, and they moved less frequently. So basically what happens is that the viraliferous whiteflies remain more time motionless and move slower than non-viraliferous whiteflies. And why did they do this? Well, after running these EPG experiments, using the technique that I explained, we could see that actually the viraliferous Bemisia tabaci whiteflies were more interested in feeding than the non-viraliferous whiteflies. Actually, they did higher number of phloem contacts, and they spend more time feeding in the phloem, and they spend longer time salivating in the phloem, which means that if they spend longer time salivating in the phloem, there is higher probability, higher risk of virus transmission. This means that the whiteflies that are viraliferous behave in a different way than the non-viraliferous, so they get more interested in feeding and more interested in salivating in the phloem, which finally increases and enhances virus transmission. So this was another major finding that we had, that also this change in the behavior of viraliferous whitefly could also benefit the vector, because they can find the phloem and spend longer time feeding in the phloem. So there is a mutualistic relationship here also. And actually the behavior of aphids and whiteflies, depending on the virus vector component, can be very different. For example, you have some viruses which are transmitted in a non-persistent manner, such as CMV, which actually attract the vector, and this is the case of CMV and Aphids gossypii. Infected plants produce volatiles that are very attractive to the vectors, while in the case of viraliferous insects, where you have whiteflies that carry the vector, that carry the virus, the healthy plants, the infected insects arrests on a healthy plant. So this is the situation of a direct effect on the right, and the situation of a plant-mediated effect on the left. But both effects, finally, what they do is that in the case of the non-persistent virus, the CMV, once the aphid starts to probe, they reject the plant as a feeding source. So this means that they are attracted to the infected plant, but then they move to another plant because they don't like to feed. And this will enhance the transmission of a non-persistent virus, because the aphid will just probe the plant and rapidly move to another healthy plant around. While in the case of the direct effects, the situation is completely different. What they do is they actually like to stay on the plant to access the phloem, the violiferous insects, and this will also promote the transmission of a circulative virus such as the tomato yellow leaf curl by having a direct effect on the vector. So what we have is a pull-push strategy that will explain how the non-persistent viruses are transmitted more efficiently, and the pull-arrest strategy to show how the persistently transmitted viruses can be transmitted more efficiently just by manipulation of the vector behavior by the own virus, either indirectly or directly. So this was another conclusion of some of the studies we conducted in our lab. So there is a manipulation of the behavior of the vector depending on the virus type or the mode of transmission, and the final point is that in both cases you get a higher spread and an enhanced spread. In the field of control of insect vectors of plant pathogens, we've been working with physical barriers and chemical barriers. Physical barriers are quite commonly used in Europe because they are good ways to manage insect pests such as whiteflies and aphids, which are common pests in greenhouses. And one of the aspects that we've been working is how we could improve this type of nets, how we can make the mesh size bigger to allow ventilation, because what you have is very high temperatures when you have a very dense net. So one solution could be to impregnate the nets with a long-lasting insecticide in a way that the whole of the net would be larger, but even so it will allow a good control of the vector. So we've been running some experiments of this type of nets that are widely used for malaria control, and we wanted to test them in agriculture, and we ran also some experiments using in the field for field crops in different situations. This is one of the experiments we conducted in our experimental farm, where we wanted to test this type of nets to prevent aphids to come inside the tunnels, and we used double tunnels. Actually, these double tunnels were the yellow net, one was impregnated with an insecticide, the other one was the control, and what we did was to release aphids between the two tunnels. The two tunnels had also cucumber plants which were infected with different plant viruses, some non-persistent and persistent plant viruses, and the aphids were released in these infected plants, and we wanted to test if they were able to cross the net and spread the disease. So in these tests, we found out actually that the cucumbers which were inside the interior tunnel were much better protected when you use the bifenthrin-treated net. Actually, this is the number of alate aphids that were able to cross the bifenthrin-treated net versus the untreated, and these were the number of plants infested by aphids in the bifenthrin-treated net and the untreated control. So here, this results in a lower incidence of both viruses when you use the bifenthrin-treated net. So this is one of the other aspects we've been testing. Then to move to the last research line which I wanted to talk about today, we were interested in how climate change, how environmental factors such as CO2 can affect aphid biology and aphid behavior. So as you know, CO2 concentration is increasing, is actually doubling in the last few years, and we are right now under very high CO2 concentration, and it's expected to rise very fast in the next years. So we conducted some studies to see how CO2 could influence not only aphids, but also how this will affect the plants where aphids were feeding. And we conducted some experiments in collaboration with our colleague in Australia, and we found out that even the behavior of aphids change when they feed on elevated CO2 grown plants. There is a higher phloem sap uptake, but even so, there is a reduction in growth rate. So aphids do actually less well or not as well when you grow plants under elevated CO2 conditions. This was published recently also, and here you can see how the fecundity of the aphids is being reduced when you grow them, when you develop them under plants that have been grown under elevated CO2. Also, the developmental rate is reduced. And finally, I would like to talk about the different projects we have right now on two bacterial diseases which are hitting Europe, Southern Europe, and we have two European Union projects which deal with new emerging and devastating bacterial diseases. Bacterial diseases were not a very big issue 20 or 30 years ago. Actually, everybody was talking about plant viruses. Now, bacterial diseases transmitted by insects are really becoming a very new, important problem, not only in Europe, but also in the Americas. We have been working in two different systems. One is the Xylella fastidiosa, which is producing a disease that you know well in the Americas, which is Pierce disease, which is a severe disease in grapevines in California. But in Europe, it's not, we were expecting this disease in grapevines, but finally it didn't appear in grapevines, it appeared in olives. It appeared in olives and it was devastating disease in olives. Actually, they call it the Ebola disease of olives, because they kill the olives quite suddenly, and they produce this disease has killed already more than 1 million olives in southern Italy. So everybody is very, very much concerned about this disease expanding to the Mediterranean, because it can be, as you can see, devastating. And we have different vectors here in Americas than in Europe. This is a tropical disease, but the vectors can be actually any insect that can feed preferentially in the xylem. In Europe, we have this spittlebug, Philenus espumarius, the meadow spittlebug, which seems to be the main vector of this disease, while in the Americas we have sharpshooters spreading this disease. The other system which we are working on is the one on Candidatus liberibacter solanacearum, which is a bacteria transmitted by psyllids, and this is a disease which is called potato zebra chip disease, which is a major disease in some places in the USA. But in Europe, we don't have it in potatoes, but we have it in carrots and celery. But we are really aware that it can jump to potatoes, and this is another type of studies we've been doing to see the risk of expansion of this disease to other crops, other cash crops. So for the Xylella fastidiosa, which is the one that I'm going to talk to you a little bit more because it's one of the major concerns right now, we did a field survey of the vectors in olives, not only olives, also almonds, grapevines, and citrus, which are the main Mediterranean crops that can be affected by these bacteria. We are studying actually now the life history of the vectors of this disease, and also we are studying the feeding behavior and the transmission of the bacteria by Philenus espumarius. And finally, we want to develop control tactics to try to reduce the spread of this devastating disease. So this is the map of Spain. Most of the olives are grown in the southern Spain, so this is where we are doing most of the surveys, also in the Mediterranean area. And these spittlebugs are found in the wheats, especially in farms that are using organic farming, the ones that are doing cover crops to prevent erosion, for example. And those spittlebugs really develop well in high populations in places where you have natural vegetation growing most of the year. And this is where we can find them, no? This would be some of the pictures where you can find most of the vectors or most of the spittlebugs in places such as this cover crop that is being used in olive orchards, in olive groves. And we've been identifying the potential vectors and understanding the biology and the life cycle of these vectors. Also, we are running some EPG studies, of course, to understand when is the bacteria transmitted, at which point which is the precise timing of the infection, how long does it take the insect to transmit the bacteria, because that could be important to find ways to control the transmission. And we've been doing different correlation studies. This technique which serves you to localize the insect stylets during specific moments. When you see a given waveform, you can do cryofixation with liquid nitrogen and then localize the stylets with this technique exactly in which tissue is the insect feeding and to understand when is the transmission happening. Also some membrane feeding studies and some video recording studies to know and understand the excretion behavior of the vector. So finally, we have this cycle that we know now in our most of olive groves where we have been studying the cycle. We have the eggs in November, February, which are laid mainly on debris and volunteers and some soil, even in the straw of the grasses that you can find below the olive trees. The nymphs developed by producing these foams during February to April. Then you have adults in these weeds, which will happen to be in April, May. But then these insects, this adult stage, can move after these weeds dry out, because as you know, olives are grown under dryland conditions, so these weeds will dry all over the summer. After May you won't have anything there. So then they will move to woody hosts, mainly oaks, and this is where they remain in the summer until they fly again after the first rains in the fall. So this is the way the cycle of the meadow spittlebug behaves in our latitude, and this is how we are trying to propose some control actions to reduce at least the numbers and the probability of transmission of the bacteria by controlling the vector. So we are thinking that the most vulnerable stage is the nymphs, which they don't move so much, and they remain in the grasses. So here you can use stillage, or you can use some biological control natural enemies against the nymphs, or you can even use herbicides to remove the insects in the precise moment. Also, you have to reduce landing rate and visits, because actually the problem with this insect is that they don't feed on olives, but they will land on the olive, they will probe the plant, and that's enough to transmit the disease and move it from plant to plant. And here we are going to test different compounds that may be effective to deter landing, such as kaolin and some other chemicals that might be effective in reducing the visits. Reduced vector numbers in the olives will not work so much because they don't feed actually in olives, but we can reduce the number of eggs by tillering or incorporating the eggs into the soil after the fall. That's another option. So just I wanted to tell you that, well, before telling you that, just wanted to tell you that it's very important that all this research that we have been conducting that could reach the media. The media in Spain now, they are much more worried with political issues, mainly independence of a region of Spain which is called Catalonia, that they want to become a new country. And of course, this doesn't matter to anybody. But we were actually very, in the prime time television 5 days ago, last Sunday actually, they emitted a video, a program during the national news about the cycle of this vector and about the work we are conducting in our lab. So this means that even politics are much more an important issue for television in Spain right now. We're able to have a 1 minute spot in the news, and this is what I wanted to show you now, that even politics are important and will be a major issue. We are able to talk about Xylella fastidiosa in olives. So this is, for me, it's a very big satisfaction that we were able to have this program in our during prime time last Sunday. That's my lab. How's your Spanish? Okay, so for me this was a very important day because actually I was traveling here the day after and they really delayed this news for 1 month because of politics. But they finally they showed up this spot, and at least we have EPGs in Spanish television and our technique and our lab there to show people that there are some other important issues in the country and in the world. So thank you very much. I would like to acknowledge Purdue with really all my heart, and I would like to tell everybody that I received excellent lessons and I am really happy to be here with you, to receive this prestigious award and to have the opportunity to give this lecture and to talk to you of all. Of course, most of the work or all of the work I've been talking today, I have to acknowledge my team at CSIC in Madrid and my students, and also, of course, my family, who has helped me a lot during all these years, my 2 sons and my wife, who is here with us, and also, of course, my family, my brothers and sisters, which have helped me during my career, and also, of course, my parents, who encouraged me to come abroad and stay here with you 2 years and learn so many things. Thank you very much.
So there's a reception after this. I'm sure there'll be plenty of opportunities to speak to Alberto about various aspects of his work, but the actual formal presentation right now is John. So, yes.
Thank you.
Congratulations.
Thank you.
Keep clapping.
Thank you.
Lovely presentation.
Thank you.
So, yeah, thank you. So there's a reception out just outside there in the usual spot. So join us and Alberto for some lovely bits and drinks and things like that. Thank you.
Alberto, you mentioned that do the plants taste different when they have a virus to the aphids? They like the virus plants better?
Yeah. Some of them are attracted and they prefer the infected plants, and in the others some other viruses are rejecting the aphid. But this means that the aphid doesn't like the plant when they feed, but they are attracted by volatiles. Means that they probe and leave. Okay. So I didn't know if there were questions or not, but anyway, I probably went out of my time. I don't know how long it lasted.
Only a little.
You remind me to get a couple extra shots just to make sure we got her covered.
Okay.
Maybe use this corner as a backdrop.
Yep.
Yeah. Here,
Okay, let's step back from it a little bit.
with or without the
Oh, I want the plaque.
You want the plaque?
Oh, absolutely.
Okay.
Upside down, mate.
Oh,
Glad you caught that.
I had to make sure because I was about to give it to him upside down before.
okay.
All right, why don't you cut it in a little bit? There
Okay.
you go,
Yeah.
like that. There you go. 1, 2, and 3. One, 2, and 3.
And one with.
You want to?
Oh, absolutely. Yes, please.
Okay.
Wait, I didn't get that. One, 2, and 3.
Thank you.
Thank you. Congratulations.
Thank you. Thank you.
Good. Good. Yourself? You guys carry on much longer?
Yeah, well, we. Like this.
Vamos a sacarle una foto.
La placa, sí. Bueno, la sacamos luego, es muy
¿Sí?
bonita.
Déjame que le saque una foto.
Sí. Beautiful.
For the family is waiting for
Beautiful.
pictures.
Beautiful. Yeah, I would like to know where to find the talk. So people is. Hi.
Alberto.
Hello.
Sue,
Nice to meet
remember?
you. Yeah, sure. Sue Cameron.
Yes.
Nice to see you.
Yep.
Nice to see you again.
Yeah.
How are you?
Sounds like you've been busy.
Yeah. Yeah, I wanted to talk to you. I told the students I want to visit to say hello to Sue before I leave. So great you came.
Yeah. Yeah,
You're
I
still
wanted
working
to get over
here.
earlier, but I wasn't paying attention at the time, and it was like, oh, it's
Yeah.
5 till 3.
Well, we had things to do all the day, but when the students came, I said, I know Sue Cameron, where is she now? And they told me that you are in a different building, in Yale or
The
Yale.
one with the greenhouse.
Okay, great.
Yeah.
So how are things going?
Going well.
Going well?
Keeping busy.
Still busy in the...
Yeah.
Great. I was remembering those days when I took that picture and I wanted to show my work counting aphids. You were there,
Yeah.
preparing all those plants.
Yep. Yeah, it's starting my busy
Yes.
time now, so I've been
With Jaime, I know you